the science behind the PET scan. Hi, and welcome back. This video series will help you understand, step by step, how a PET scan works. Make sure to watch the videos in the correct order, as the information in each video builds upon the previous ones. Here you can see the journey we are on to understand the science behind the PET scan. In the previous video, we talked about radioactive decay and half-lives. In this video, we'll learn about how the radio tracer is created. Next time, we'll look at the creation and analysis of the images. The other videos are linked in the description box down below, so if you've already seen this one or you've skipped the video, make sure to check out the others. Today, we'll talk about the most important step before a scan, the synthesis of the radioactive tracer. Once again, we're starting off with a short repetition. Next, we'll look at what the radioactive tracer does. Then we'll talk about the cyclotron, where the radioactive isotopes are produced, followed by the synthesis of the radioactive tracer itself. And finally, we'll summarize the most important concepts. An unstable nucleus can remain unstable for a long time, but it will eventually decay into a stable nucleus. The decay relevant for PET scans is the beta plus decay, during which, due to the laws of conservation, a positron and a neutrino are emitted. The positron is the particle that is needed for PET, as mentioned in the name, positron emission tomography. The half-life is the time it takes for an initial amount of an isotope to decay into half that amount. The half-life of an isotope can range anywhere from nanoseconds to billions of years. Look at these impressive scans of a brain. We are already one step closer to finding out how they are created. Let's dive in. How do our organs even become visible on the scan in the first place? Well, that's exactly what the radioactive tracer is used for. The tracer is injected into the bloodstream of the patient in order to highlight the body parts, or even the specific cells under examination. The radio tracer is made up of two main parts. The first part is a radioactive isotope. The second part, a biological molecule. The fascinating thing about creating the radio tracer is that it can be tailored to a substance that the organ would use naturally. When creating the radio tracer, a biological substance that the organ will very likely take up is selected, and a radioactive isotope is added to the substance. This process of adding a radioactive isotope to a biological molecule is known as radiolabeling. Let's look at a specific example of tailoring a radio tracer. Imagine the doctors are scanning for a tumor. Cancer cells absorb more glucose than normal cells because cancer cells replicate much more quickly. They get their energy for the fast replication from the glucose that they absorb and turn it into ATP. In order to highlight the cancer cells, a tracer containing a radioactive isotope and a glucose-like molecule is used. When the tracer is injected into the patient's body, the tumor will gobble up the tracer because it is feeding on the glucose. After that, the tumor will be highlighted and become visible on the scan. The tumor is highlighted because the radioactive isotopes start decaying, and special cameras are able to pick up the gamma rays that are emitted. We'll talk about the cameras in the next video, so keep watching. The first part of the radio tracer, called the radioactive isotope, is typically produced in a cyclotron. A cyclotron is also known as a particle accelerator. In a nutshell, the goal of a cyclotron is to accelerate a charged particle to high speeds, at most, typically a third of the speed of light, and to shoot that high speed and high energy particle at a stable isotope. As a result of the charged particle colliding with the stable isotope, the isotope becomes radioactive. In general, a charged particle refers to either a charged atom, known as an ion, or a charged subatomic particle, for instance, a proton. It is very important to note that the cyclotron has limitations. It can't accelerate neutral particles, electrons, or positrons, because neutral particles have no charge, and the masses of the latter two are too light. So, when we talk about a charged particle with respect to a cyclotron, that means a proton or an ion. A cyclotron consists of the following: 2 D-shaped hollow metal electrodes, called Ds, a vacuum chamber that encases the Ds, an alternating voltage source, and 2 magnets, one above and one underneath the vacuum chamber. In order for the magnets not to cover up the entire illustration, I made them transparent. The vacuum chamber has a specific entry where the charged particle is injected, and exit tube where the charged particle is shot out of the chamber, but these are not shown here. Most importantly, an electric and a magnetic field are created. These fields are the key element to a cyclotron and how the particle is influenced. In short, the electric field, depicted by the horizontal red lines, is responsible for the acceleration of the particle, and the magnetic field, depicted by the circles, is responsible for the path that the particle moves along. Let's follow a particle around the cyclotron. The charged particle is injected into the vacuum chamber and placed in the gap between the 2 Ds that are facing each other with their flat sides. The 2 Ds are constantly alternating between a positive and a negative pole due to the voltage source that they are connected to. In other words, when one D, let's call it D1, is negative, the other D, D2, must be positive. Because of this alternation of poles, an electric field is created in the horizontal gap between the 2 Ds. This electric field accelerates the particle. Going back to our example where the charged particle was placed in between the Ds. Because of the electric field, it is accelerated towards the first D. As soon as it has reached the first D, its path curves due to the magnetic field that is perpendicular to the planes of the Ds. If it weren't for the magnetic field, the particle would go right into the wall of the vacuum chamber. Because the magnetic field does not change the particle's energy, the modulus of its velocity stays constant whilst it is passing through D1. As soon as it has traveled through D1 and reaches the gap between the Ds again, it is influenced by the electric field, where it is once again accelerated. Due to the increased velocity, the particle's path will now have a larger radius when it reaches the second D. If we continue this pattern, the path that the particle moves along is an outward spiral. When the particle's path can no longer increase in radius, the high-speed, high-energy particle is shot out of the vacuum chamber, where it collides with a stable isotope. This collision of the high-energy particle with the stable isotope causes a nuclear reaction, and it becomes a radioactive isotope. The trajectory would look like this. And the collision followed by the nuclear reaction, would look like this. Here are some radioactive isotopes that are used in PET scans. F18 is an isotope of fluorine. It has a half-life of 109 minutes. We already talked about this one in the last video, when I introduced you to the concept of half-lives. C11 is an isotope of carbon. Its half-life is 20 minutes. N13 is an isotope of nitrogen and has a half-life of almost 10 minutes. O15 is an isotope of oxygen. Its half-life is a bit over 2 minutes. As you can see, they all have relatively short half-lives. There are pros and cons of having a short half-life. One problem is that the radioactive isotope needs to be produced on site. If it were produced externally, it would have decayed almost completely by the time it reached the PET scanner. This calls for having special equipment on site, for instance, a cyclotron, which is very expensive. On the other hand, having a short half-life means that the radiotracer won't stay radioactive in the patient's body for a long time. This means that the risk of experiencing long-term health problems from the radiation are minimized. In addition to minimizing the risks, having a short half-life also enables the doctors to safely give the patients a high dose of the tracer. A high dose means that the radioactivity is stronger. Therefore, the PET scanner will pick up a stronger signal, resulting in an image with a higher contrast. It is important to note that a high dose does not imply a large quantity. For PET scans, the actual amount of the tracer that is injected is tiny, but the concentration of the tracer is very high. After the radioactive isotope has been produced, a specific molecule needs to be added in order for the radiotracer to be complete. The doctors decide on what molecule to add depending on what organ is under examination, while avoiding anything the patient is allergic to. Let's take a closer look. As previously mentioned, F18 is a very common isotope used for the scans. It is combined with a molecule called deoxyglucose to form F18 fluorodeoxyglucose, short FTG. This tracer is often used in the field of oncology. Deoxyglucose is a glucose analog. That means it has a similar structure to a glucose molecule. On the left, you can see deoxyglucose, and on the right, FTG. To get FTG from regular deoxyglucose, the hydroxyl group at the C2 position is replaced by the positron-emitting radioisotope F18. The radioisotope is circled in red. This is a perfect example of radiolabeling. Other molecules that are used for creating radiotracers include H2O, NH3, CO2, and O2. Another example of a radioactive tracer is N13 ammonia. That means the molecule ammonia, NH3, is radiolabeled with the radioisotope N13. The radioisotope is circled in red. N13 ammonia can be used for myocardial perfusion imaging, for instance, which is a procedure that measures the blood flow to the heart muscle. Due to its relatively short half-life, it needs to be produced on site. The image on the right shows you the scans of a heart muscle. The upper row shows the heart under stress, and the lower row shows the heart at rest. The brighter areas are where the heart absorbs the most tracer, highlighting the areas with the highest blood flow. The stress image here shows areas where there is weak uptake of the tracer. The heart we see here has a higher blood flow at rest than at stress. Let's sum everything up. A tracer consists of a radioactive isotope and a molecule. A cyclotron accelerates particles to high speeds and shoots them at a stable isotope, creating a radioactive isotope. The most common tracer is FTG, where deoxyglucose is labeled with F18. A short half-life comes with pros and cons. For instance, minimizing the health risks, but also requiring on-site production. A little self-test. Try recalling what the two main parts of a radio tracer are, and why it is so important to tailor the tracer. Thank you so much for tuning in, and see you soon!